Hello and welcome to Nextopia Medical Lectures. In this session, I shall be discussing about embryology. We'll start discussion of anatomy. In anatomy, first we'll discuss embryology, then histology, then we'll go to anatomy proper. Please remember that anatomy is different from physiology because the fact that anatomy is purely the regional study, like region wise you have to study. If I say upper limb, then you will have to restructure or reconstruct the upper limb as if you are seeing the patient in front of you, and then you have to imagine the muscles, the bone and all right. If my voice is not clear, if my video is not clear, then please inform me. Immediately I'll change the settings. My internet was showing a lot of problems today. I apologize for that. This is a new link. I've started the class now. So in today's class, we shall be discussing about the introduction to embryology. What is embryology? We'll try to find out. Then we'll discuss what is an embryo, what do you mean by fetus, what do you mean by a newborn baby, what do you mean by how is the embryo formed, what is the cell that is responsible for fertilization, what are the different types of growth and development changes that we are going to expect in this fertilized zygote. All right, and then we'll also discuss how the cells divide and how is the growth and development taking place. So in this chapter, the chapter is very simple, but it has got few important theoretical applications, like understanding the definition of embryology. So first thing is, what is embryology? Embryology is a study of development of an individual human being before birth. Development before birth is embryology. So the crux of embryology, if you ask me, sir, what is the crux of embryology which I need to remember for exams, you need to remember the timeline. In the first week, what is the development? Second week, what is the development? From first to fourth week, 4 weeks, what is the development? For first 3 months, what is the development? Second 3 months, what is the development? So you will have to understand these points, and then you will have to correlate these points. And there are certain congenital disorders which occur because of failure of proper embryological mechanisms or embryological life cycle. So those problems should be addressed by a clinician. All right, if you understand this normal embryology. The second application of normal embryology is understanding the pediatrics, the study of diseases in children. The third important application is you understand gynecology, to understand infertility chapter. Especially the first few weeks of pregnancy are very, very important to understand infertility chapter. So all these I'll be dealing subsequently. In this first chapter of basics and introduction, the study of development of a human being before birth. Before birth, I can also call it as prenatal. So by this, you know what is postnatal. The postnatal is after birth, is it not? So this is the definition of embryology. Now let us try to understand what is the structure you are calling as an embryo. What is an embryo? An embryo is a developing group of cells in the first 2 months or 8 weeks of intrauterine life. 2 months, first 2 months of life, or first 8 weeks, first 2 months or first 8 weeks of intrauterine life. All right. Then what is a fetus? Fetus is defined as the developing human being from the 3rd month or 9 weeks from the third month or from 9th week till birth. So this is a fetus till birth. I'm sorry. Then, so development before birth you are calling it as prenatal development. The development after birth you are calling it as postnatal development. Please remember that 90% of the organ development has occurred. 90 to 95% of organ system development will occur prenatally. So that is how prenatally first 3 months are so important. The first 3 months, that is embryo, the first 2 months are so important that most of the important organ systems are already completely functional by the end of 8th week. All right. So you need to understand that this 2 months of pregnancy, first 2 months of pregnancy is so important for us. That's the reason why the supplements should be given properly. There should not be any deficiency of minerals, carbohydrates, proteins. The person who is pregnant should not be fasting, should not be starving. I'm not talking about the routine fast of religious fast that is kept, that is intermittent fasting is okay. I'm talking about starvation, like people who completely deny food, eating food. That is not advisable. So please remember that prenatal development is divided into pre-implantation, embryonic phase or embryo, then the fetus. So few days before implantation, that is from the, say suppose 14th day of menstrual cycle, 12th to 14th day of menstrual cycle, ovulation has occurred. From this point, 12th day of menstrual cycle, ovulation has occurred. From this point of time, fertilization occurs within 72 hours after ovulation. Within 72 hours after ovulation, fertilization occurs. So from this point of time till the implantation, you are calling it as pre-implantation. Implantation of an embryo. I'll discuss what is implantation, everything in my subsequent classes. After that, I already told you that first 2 months is an embryo and fetus is from 3rd month till birth. So from here, if you see, the fetus undergoes birth. Then after birth, the fetus now, it is called as a neonate. The life of a neonate is 28 days. First 28 days, the newborn baby, you are calling it as fetus. First 28 days after birth, you are calling it as neonate. Again, we have got early neonate, late neonate. We'll discuss that in pediatrics. All right. So this is the life cycle of the fetus. So implantation is the first step. So before implantation, there are certain changes that are occurring in the uterus. We call it pre-implantation. So from pre-implantation to implantation. Immediately after implantation, it is called as an embryo. And after that, it is called as a fetus. And now the fetus is, after birth, it is called as a neonate. All right. How does this occur? This occurs because of, thanks to the presence of gonads in both male and female in the form of testis and the ovary in male and female respectively. So that male gonads, that is testis, produces spermatozoa. The female gonad, that is ovary, produces the ova. When spermatozoa or sperms and ova, that is after ovulation, the ovum, both of these, they undergo fusion, fertilization has taken place. we call it fertilization has taken place. Remember the fertilized ovum, now it is called as a zygote. This zygote should undergo implantation in the uterus. After implantation, the embryo is formed and the embryo undergoes development. This is what we are studying in our classes. All right. In infertility chapter of gynecology, I'll be teaching all these stages of development of spermatozoa. I'll discuss that also now, but I'm talking about diseases. I'll discuss diseases in infertility chapter of gynecology, the development of spermatozoa, how to treat the spermatozoal disorders, then ovum disorders related to infertility because of problem in the ovary or ovum in this flow chart, or problem related to fertilization, or problem related to zygote formation, or problems related to implantation. Like that we'll divide and we'll read, so it becomes very easy for us. But before that you need to understand. Now one question you may ask me, sir, how are the testes and the ovary, the two gonads, are producing the spermatozoa and ova? These are called as gametes. All right. How is a gamete produced? So the process of production of gamete is called as gametogenesis. Gamete is nothing but it is the sex cells. So production is called as gametogenesis. All right. So the process of production of gametes in the gonads, testes or ovaries, so we call it gametogenesis. So we have got two types here. So we have got spermatogenesis. We'll read this in detail. A lot of questions are asked here. Steps of spermatogenesis. Then we'll also discuss oogenesis, that is production of the ovum. Then after that, fusion of this sperm and ovum, called as fertilization. We'll discuss that also. Then after that, growth and development occurs. So please remember, once a life cycle is complete, we'll understand how the life cycle propagates. So these are the important basic qualities of a living organism. A living organism, apart from growth and development, the most important property of a living organism is, the first one is protection. That is, you should be able to protect, the human being should be able to protect himself or herself from harsh weather, from various other issues, and then propagate the life. This is first important. Second thing is growth, that is proper growth from a child to adolescent, from adolescent to the adult, from adult to old age. So these are all the physical development. Then mental development, that is intelligence, social behavior, how you interact with others. Everything should be learned, and this is what is a basic property of a human being. After that, the most important property, the third property, is propagation of species. Propagation of species means we should be able to produce gametes, so that the species is propagated, and this is how the population grows. So for this, what is essential is, for propagation of species, the most important thing is reproduction. All right, reproduction is a mechanism by which we produce new generation, and that is brought about by the internal sex organs which produce the gamete. Each gamete, that is each gonad, produces a separate set of gametes depending on the sex we are talking about, male sex or female sex. So spermatogenesis and oogenesis together, they are referred to as the gametogenesis. All right. So for the development of a human being, we have divided, I already told you, the events into pre-implantation, implantation, then embryo, then after that the fetus, and then the neonatal life, that is postnatal life or neonatal life we call it. Please remember, the embryonic period is extending from the so in this flow chart, if you see first 2 months, if I say first 2 months, it is from the third week to 8th week, actually third week to 8th week. So first 2 weeks is in pre-implantation. First 2 weeks is implantation or pre-implantation phase. Then from the third week to 8th week is embryo formation, and from the 9th week to 9 months, 9 week to 9 months, 9 week to 9 months, it is the development of the fetus. We call it fetal period. 9 weeks to 9 months is the development of fetus. Each of this phase has few important steps which you need to remember. For example, in case of pre-implantation, what are the important steps? What are the important features of pre-implantation phase? Let us see. What are the important steps of pre-implantation phase? So the pre-implantation phase includes, as I told you, the first is fertilization. You can write SATION or ZATION, whatever it is. Then this fertilized zygote undergoes cleavage. Cleavage is division of a fertilized zygote into 2 cells like this, from 2 to 4 cells, from 4 to 8 cells, from 8 like that to 32 cells. This 32 cell stage is called as blastocyst. So from cleavage of this zygote to morula followed by the blastocyst. This blastocyst is a structural and functional specialization and reorganization of cells. The cells of blastocyst, you are calling it as blastomeres. So important point about this is blastomeres have got structural and functional specialization. Then the blastocyst undergoes, the next step is implantation. All right. So fertilization, then cleavage, formation of blastomeres or blastocyst, then implantation. Implantation is a process of attachment of these cells, bag of cells or mass of cells. It is in this stage of blastocyst implantation occurs. For example, if this is the uterus, these are the fallopian tubes. Fertilization has occurred in the fallopian tube. From here the zygote travels and gets implanted mostly in the posterior wall of the uterus, endometrium, innermost lining of the uterus. And the stage of implantation is attachment to the posterior wall of the uterus. This stage, at what stage it is getting implanted? It is at the stage of blastocyst. It's an important question. Blastocyst. The implantation is a process of attachment of blastocyst to the uterine endometrium, mostly occurring in the posterior wall of the endometrium. All right, at the level of the fundus of the uterus. All right. Now what happens is the blastocyst undergo differentiation and specialization into different types of cells. So we are calling it as differentiation of this blastocyst. See, blast means what? Why are we calling it blast? We are calling it blast because it has a property of a stem cell. A stem cell is called as a blast. It has got a property of giving rise to different structures. Again, it is divided into embryoblast and trophoblast. Embryoblast and trophoblast. Trophoblast is again divided based on the type of cell into syncytiotrophoblast and cytotrophoblast. See, today is my first class on embryology, so I am not going into details of syncytio and cytotrophoblast. I'll discuss everything. I'll draw all the diagrams and I'll show you how the syncytiotrophoblast and cytotrophoblast differentiate to form different organs and how the blastocyst gets converted into the next stage of during implantation and following implantation. All right, so this is all about the pre-implantation phase. Next comes the embryonic period. In case of embryonic period is from 3rd week to 8th, 3 to 8 weeks. This is very important. Third week to 8th week. Here what happens is the embryonic disc, now we are calling it as a germ disc. germ disk. This is undergoing differentiation into triple layers, 3 layers. So we are calling it as trilaminar, 3 layers. Laminar means layer, tri means 3. Trilaminar germ disk, trilaminar germ disk. And this you are calling it as the ectoderm, the endoderm, and in between we have got the mesoderm. This differentiation of embryonic disc into the 3 layers is an essential, most important aspect that is occurring in the 3rd week. From 3rd week this is starting. After this, what happens is, once you differentiate 3 layers, ectoderm, mesoderm and endoderm. Ecto means outer, meso means middle, endo means inner, is it not? So 3 layers are formed. Now these layers will get transformed into organ development. Like for example, heart. If you see heart, the ectodermal component of heart will be the outermost layer of the heart. The mesodermal component of the heart will be the muscle of the heart. The endodermal component of the heart will be the innermost lining of the heart, something like that. I'll discuss that when I'll teaching cardiology development. I'm just giving you an example. All right. So after this, what happens is, now see, when organ is developing, we also need some extra supportive structures, like we need clothes, supportive structure, like we need clothes to cover ourselves. The embryo also needs some layers of connective tissue to cover itself. That is called as extra embryonic supportive membranes. Extra embryonic membranes. Now these membranes have different properties. They also help in development of organ and organ system. They protect the embryo, they protect the different organ systems, and they also help in circulation. They also help in preventing any disease from entering into the fetus. It also prevents autoimmunity disorders and all, everything in molecular level, if you try to understand. So this is termed as extra embryonic membrane. Extra embryonic membranes. Now what are the examples of this extra embryonic membrane? We have got the placenta, the umbilical cord. See, each and every structure I'll discuss in detail. The placenta, the umbilical cord, the amnion, the chorion, which is a part of placenta again, and the allantois. The amnion, the chorion, the allantois, and the umbilical cord. This is all about the development of embryonic development. So embryonic development divided into the first phase of differentiation into 3 layers. After that, organogenesis. Organ development, you are calling it as organogenesis. After organogenesis, along with organogenesis, not after. Along with organogenesis, we need some protection to the organs, so various membranes are formed to protect the organs. Now once the organs are formed, this is happening till the 8th week. After that starts the fetal period. The fetal period starts after that. From 9 weeks to 9 months. From 9 weeks to 9 months, the fetal period starts. And this fetal period is responsible for growth of the fetus in all the dimensions, longitudinally, horizontally, 3-dimensionally, all the horizons, and specialization of various body structures. Now once the organ system is formed, once the organ is formed, we need to specialize it and few corrections in the organ system so that the normal development is carried out. And this is how the human body is completely formed at the end of 9th month. After that, obviously the postnatal development we need not discuss. The neonatal period, the postnatal. After that, what happens after fetal period? The postnatal life, which includes the neonatal period, again divided into early neonatal life and post-neonatal life. The neonatal period. Then after that, infant up to 1 year. So this is up to 28 days after birth. So from 1 month to 1 year, from 1 month to 1 year, you are calling it as a infant. After that, childhood starts. Again, childhood divided into early childhood, the preschool, the infant becomes toddler, then from toddler it becomes, so parts of childhood, divisions of childhood, we have got toddler, then we have got preschool age group, then school going age group, then after that we have got adolescent, then adult, adult again we have got young adult, old adult, like that. So this is how is the life cycle of a human being. So in embryology we are dealing with the processes and regulations of the prenatal growth. We are not bothered about the postnatal growth in case of embryology. The life of the fetus starts in the fallopian tube and it ends after the immediately following the delivery. Embryology includes the integration of various complex molecular, cellular, structural, hormonal processes which occur in the first 9 months of the first 9 months of human being, especially the intrauterine life. Now there are various specializations in embryology. First we'll discuss general embryology. After that we'll discuss the systemic embryology. In systemic embryology we'll discuss various systems. In systemic embryology we'll discuss various systems, and those systems. After that we'll discuss teratology, that is branch of embryology which deals with abnormal embryonic development following or birth defects. We call it teratology or teratogenesis. Then after that we'll be discussing the clinical implications or clinical applications of embryology. This is all about the introduction. So what are the important basic processes of growth? The basic process of growth is, first is multiplication, then differentiation, then organization. All right. So first is growth includes multiplication, then differentiation, then organization. So if you go into properly classifying things, we have this. After organization, then we talk about the stem cells. What are the properties of stem cells? So stem cells, it is nothing but it is a self-renewable differentiating cell of our body. It's an undifferentiated cell. A stem cell is an undifferentiated cell, or you can call it a differentiating cell. Undifferentiated cell or a differentiating cell, and which has got two types. Actually, most of the stem cells are embryonic stem cells. In the embryo life, only 90%. Few stem cells are adult stem cells, or we can also call it somatic stem cells. So what are the important properties of adult stem cells or somatic stem cell? Is that they are tissue specific. For example, stem cells present in the bone marrow, they're specific to produce bone marrow related blood cells, like that. So they are tissue specific. It's an important property, and they are very, very limited, and they remain throughout the life of an individual. And in some people whom they have got some genetic disorders, we can give stem cell therapy, which will cause regeneration of or replacement of the deficient cells and treatment of blood disorders. All right, this is the clinical application of stem cell. So what is the basis of stem cells? The basis of stem cell, the basic property of stem cell is self-renewal. Suppose you have lost few cells from your body. Suppose you have lost few cells of your liver. Though the liver should have an ability to replace those cells back, and we are calling such cells of liver or the lungs or whatever it is, organs, which grow back or self-renewal and differentiation as stem cells. Stem cells are named according to their property. So depending on their potency, depending on the cell potency, stem cell potency, the stem cells are divided into totipotent cell, then pluripotent cell, then multipotent cell. So multipotent cell. All right. What is a totipotent cell? A totipotent cell is a cell type in the embryo which, in addition to extra embryonic or placental cells, all right, so we have got extra embryonic cells, is it not? And we have got placental cells. So in addition to this placental cells and in addition to this extra embryonic cells, the stem cells which are present, these are the totipotent cells. The embryonic cells within the first couple of divisions, that is, say suppose the zygote has divided into 2 cells, then into 4 cells, I told you, then into 8 cells like this. So immediately following first few divisions, immediately following fertilization, so these cells are totipotent cells. All right, the cells which are formed immediately following fertilization, these are the totipotent cell. So we do not know which cell has got an ability to transform into an organ now at this stage of immediately following fertilization. So you are calling it a totipotent. For example, the cells of, so what is the example of totipotent cell? For example, the cells of blastocysts, all right, or blastomere or blastocyst is a totipotent cell. Next comes a pluripotent cell. Pluripotent cell is a type of cell which gives rise to all the different types of cells present in the body. All the different cells. So it has got an ability to produce, it produces, it can give rise to all the different types of cell types, all the cell types in the body. Hence, we are also calling it as an inner cell mass. So we'll divide this blastosmear into inner cell mass and outermost covering of trophoblastic cells. So this is inner cell mass, which is giving rise to the pluripotent cell, which has an ability to give rise to all the different cell types. All right. Multipotent cell means a stem cell which has an ability to produce, multi means many, is it not? Multi means many. So more than one cell type. You may ask me, sir, what is the difference between a pluripotent cell and a multipotent cell? Pluripotent cell give rise to all cell bodies, all cell types, whereas the multipotent cell give rise to one and more than one. But it is limited. It is not all. So it has got a limitation. It is limited to few different cell types. That is, you are calling it as a multipotent. For example, the mesenchymal cells of our body. The mesenchymal cells of our body. The mesenchymal cells of our the cord blood stem cells, or the bone marrow cells, the bone marrow stem cells present in the bone marrow, you are calling it as a hematopoietic cells. So this is how sometimes we are able to harvest the stem cells, and based on their property, whether it is totipotent, pluripotent or multipotent, you can simply use it in certain clinical conditions. Now apart from this, we have got a category of stem cells which can develop into cells of only one category. So the stem cells which produce only one cell type, what do you call it? The stem cell which produces only one subtype, you are calling it as oligopotent. The stem cell which can produce one cell type, you're calling it as oligopotent. Here one example is, you can also call it as unipotent. All right, unipotent or oligopotent. All right, so there is a slight difference between unipotent and oligopotent. I'll tell that now. So what is a totipotent cell? Totipotent cell has got a capacity to differentiate into embryonic as well as extra embryonic cells. Embryonic as well as the extra embryonic cells. What is a pluripotent cell? All the cell types of embryonic body develop from pluripotent cells. What is a multipotent cells? More than one cell type is a multipotent. What is a oligopotent cell? Only one category of cell is produced, whereas only one category of cell is produced. So I write here, produce only one category of cells. Category means either related to one organ system. Like for example, bone marrow stem cells. Only bone marrow is produced, you're calling it as oligopotent. Whereas unipotent means the ability of a stem cell to produce only one type of cell. Only one type of cell. For example, liver. For example, any organ we're talking about. So this is all about classification of stem cells. Stem cells are of different types. We have got totipotent cell, pluripotent cell, multipotent cell. Then we have got oligopotent cell, a unipotent cell. I've given you the examples. So with this, I complete the chapter 1 of embryology. In the next class tomorrow, we'll discuss the further chapters as given in the timetable. Any doubt, please feel free to ask. I'll be happy to help you out. Thank you so much.